Reversible covalent modification. In our body, we have greater than 500 modifiers of enzymes. Think of them as hats an enzyme can wear, different hats an enzyme can wear, okay? So enzymes have 500 hats they can wear. The most common hat or the most common modifier is PO43-, which is phosphate, right? And we also have specialized modifying proteins, or let's say special hats that the enzyme likes. They are called ubiquitin and sumo. Okay? So enzymes have modifiers. They have a lot of modifiers. The most common one is PO43-, and the specialized ones are ubiquitin and sumo. So these are other examples that were found. Most of them end with YL, so I would definitely know these and these and these. But if your professor emphasized on these, then you should study all of them. Explanation of phosphorylation. So what is phosphorylation and where does phosphate come from? Phosphate comes from a molecule called ATP or adenosine triphosphate. Okay? So what happens is that adenosine triphosphate donates one of its phosphates to an amino acid residue in the enzyme. This residue is in the enzyme. It donates a phosphate to this amino acid residue and it becomes ADP. What's the difference between ATP and ADP? Triphosphate means 3 phosphate molecules. Diphosphate, it means we have 2 phosphate molecules because it donated one of them to the amino acid residue. So when we say that something can donate phosphate to another group, it is called a kinase, as I have said before, right? The enzyme is reversible. How? Or not the enzyme is reversible, actually. The addition of phosphate is reversible. How is that? Because we have other enzymes that are called phosphatases. Just like we have kinases, we have phosphatases. Kinases are the ones that add phosphate to the group, and phosphatases are the ones that remove this phosphate from the group. Okay, so phosphate is for PO43-, and ases is cleavage or removing. So they are less specific. Why? Of course they're gonna be less specific because that's better. The lesser specificity they're gonna have, the more enzymes they can phosphorylate. So kinases, they add PO43- to a lot of enzymes. They are not specific and they can act on a lot of proteins. So this is actually good for us because if they're specific, then a lot of functions in our body won't be happening as we speak. Now, an example from your lecture is glycogenolysis regulation. So what is glycogen? Glycogen is the storage form of glucose. So you eat glucose and your cells take this glucose and use it for cellular respiration. And if you have excess glucose, what happens is that it goes to the liver and it's stored. It's stored like building blocks over each other. These blocks of glucose are called glycogen. Okay? Glycogen is the storage form of glucose. Now, lysis is breaking down of this glycogen, which means that this glycogen is gonna be broken down into glucose again. And it's regulated. Sometimes I need to break it down, sometimes I don't need to break it down. Okay, now how is it regulated? It's regulated by 2 enzymes called phosphorylase and synthase. Glycogen phosphorylase and glycogen synthase. Now, to explain exactly how this works is that, let's say you've just ate. Postprandial, you've just ate. We're gonna have high glucose levels, and this stimulates a hormone in our body called insulin. Insulin comes, it takes the glucose from our blood and delivers it to cells, okay? And at the same time that it's delivering these cells, it activates something called phosphatases. What did I say phosphatases were? They cleave phosphate groups. They're the exact opposite of kinases. So these phosphatases, they go to these two enzymes and they remove their phosphate groups from each of these enzymes. What happens? Glycogen synthase, when you remove one of its phosphate groups, it becomes activated. And when it's activated, it starts building the glycogen. It starts putting the glucose as these building blocks, and glycogen is synthesized. However, glucose phosphorylase, this one, when you remove one of its phosphate groups, it becomes deactivated. It's deactivated because the job of this one is to break down these building blocks. So if it's activated, then all what we're doing here is gonna go to the trash can. So this one should be deactivated so that we can build the glycogen and store the glucose that we just ate, right? Now, the second case is that you're fasting and you actually need glucose. Your body needs glucose. So what's going to happen is that you're going to have to deactivate this enzyme and activate this one. Because when you activate this one, glucose is released. And when you deactivate this one, you're not using glucose anymore to build glycogen. In fact, you are releasing this glucose into the blood so that the cells can benefit from it. So what happens is that you have decreased glucose, and this stimulates another hormone that's similar to insulin, but has the exact opposite action of insulin. It activates kinases. Insulin activates phosphatases. This one activates kinases. It phosphorylates, or it adds a phosphate group to these two enzymes that we just talked about. And when this one is deactivated, it cannot make glycogen, which means that the glucose is not being stored like this. It's being released into the blood because the body needs it. And this one is breaking down the glycogen. It's breaking it down so that it can go to the blood and the cells can take up the glucose. Okay, now we're gonna know exactly how glycogen synthase is being phosphorylated. Glycogen synthase is this one that we've been talking about, and the kinase example in this case is GSK. Okay, so think of glycogen synthase as a road, and GSK is the car that's driving over the road. So GSK actually attaches itself first to glycogen synthase at the +4 position at the serine residue. That's the first step it does. And then it starts moving. So at the first position, which is 0 at the serine, it phosphorylates it here. Then it moves forward and it phosphorylates it at the -4. Then it moves forward and it phosphorylates it at the -8, and so on and so forth. So that's exactly how it works, chemically speaking. So moving on to Zymogen activation. Proteases are zymogens. The word zymogen means that zyme actually means enzyme in Greek, and gen means genesis, which is the making of something. So first of all, why are proteases zymogens in the first place? Like, why are they pre-made and stored to be used later on? Because proteases are actually very dangerous if they're not controlled. As I said before, protease stands for protein and breaking down. So these enzymes break down proteins. And if you think about it, if you have proteases just laying around in your body, wouldn't that cause tissue damage? Of course it would. So they are proteases, they're synthesized first as an inactive form, and then when they're needed, they are activated. It's like when you store something in your cupboard and you need it when you, for example, you're cooking something, okay? So number 1 is for safety, to prevent autodigestion. For example, trypsin is in its inactive form called trypsinogen, right? Now, let's say it's overactive. What's going to happen is that it's going to start eating the pancreas, because trypsin is released from the pancreas. It looks like this. This is the pancreas. And when there's trypsin excess, it's gonna start eating the pancreas, and this is called auto because it means self. Digestion is, I think you know what that means. So proteases are only activated when needed for safety reasons. Now for speed, if you think about it, when you have something stored in a closet and you have something to do right now, you just go grab it and use it. You use it immediately. You don't have to go and prepare it and then use it immediately, right? So for speed, they are pre-made and stocked for when they're needed in bulk, like after eating a protein-rich meal. As I said, proteases, they break down proteins, right? So let's say you have just eaten a protein-rich meal. Your body's not going to start synthesizing proteases. It just activates the proteases it had already synthesized before and they've been stored. Okay, so for safety, check. For speed, check. Now for control. So it's like what I said before. Proteases are also needed for blood clots, for the removal of blood clots. So the body controls how much proteases are activated or deactivated according to its needs. An example is the trypsinogen that I talked about. So you eat a protein-rich meal, it reaches the duodenum. The duodenum is the upper part or the first part of your small intestine. And when it reaches the duodenum, trypsinogen is activated and it becomes trypsin. Trypsin then activates the other proteases, chemotrypsinogen, proaminopeptidase, and procarboxypeptidase. Now, the body, of course, has a guard that guides trypsinogen because, as I said before, the more activated the trypsin is, the more we have a risk to have autodigestion. So we have something called pancreatic secretory trypsin inhibitor. So this is a guard that jumps in and deactivates extra trypsinogen to protect the pancreas. So what is short-term and long-term regulation? Let's say for short-term regulation, you haven't been eating protein, okay? And trypsin is active. So what happens is that we either have allosteric or covalent deactivation of trypsin, and these are rapid adjustments, okay? So, for example, let's say you have eaten and you need to activate trypsin, so you have allosteric and covalent regulation that I have explained before. Now, what is long-term regulation? Long-term regulation needs a lot of time. For example, let's say you work out at the gym and during recovery, muscle recovery, your body has to increase its enzyme synthesis. So it's going to take your body hours to synthesize all these enzymes, all these proteins that are necessary for your recovery. So this is more of long-term regulation. It's not short-term. It's not pre-made, prepared for when you need it. You need time to build these enzymes and recover. So short term, it's something that is already there. You use allosteric or covalent regulation to regulate the enzyme. In long term, you increase the synthesis of new enzymes because you're building something new in this case. But in this case, you're not actually. You're just activating or deactivating an enzyme according to what your body needs, just like this example that I said here. If you need trypsin, you activate it. If you don't need it, then you don't activate it. So, what are isoenzymes? If you remember in the beginning of this lecture, I explained what isomers were. Isomers are the exact same thing as isoenzymes, but the difference is that we're talking about enzymes here. Okay, so iso means same, and enzymes are what we're talking about. So these are different forms of the same enzyme that catalyze the same reaction. But if they're the same form, then what's the difference between them? The difference between them is their properties, where their location is, their kinetic properties, kinetic as in motion, Km, Vmax, etc., and what cofactors they need to function. So we have an example here to make it easier for you to understand. So the first example is glucokinase and hexokinase. These two enzymes are isoenzymes. They are both 6-carbon molecules. The difference between them is, as I said before, their location, their kinetic properties, and the cofactors they need to function. Okay, so both of them have the same job. Both of them do the same thing. They add phosphate to glucose, and glucose becomes glucose 6-phosphate, right? This is the first example. The second example is LDH, lactate dehydrogenase. So what does lactate dehydrogenase do in the first place? Lactate dehydrogenase converts pyruvate to lactate. These two are also related to cellular respiration. So what happens is that LDH has isozymes. Just like these two, they're isozymes. LDH has different forms of itself. All of them are formed of 4 tetramers. Tetra means 4. Okay? All of these isozymes are formed of 4 polypeptide chains. Okay? But the difference between them is that some of them are related to the muscle and some of them are related to the heart. For example, LDH1, the first form of LDH, has 4, as we said before, polypeptide chains that all of them are related to the heart. We have 4 H's here, heart. For example, LDH2 has 1 polypeptide chain that's related to the muscle and only 3 related to the heart. Okay? LDH5 has all related to the muscle. LDH3 has 2 heart, 2 muscle. LDH4 has 3 muscle and 1 heart. So what we conclude from this is that LDH1 and LDH2 are mainly concerned with the heart and RBC. This is their location and where they work. LDH4 and LDH5 are related to the liver and the muscle. And LDH3 is related to the brain and the kidneys. Now, as we said, they have a different location, check. Now, what about their kinetics? So, as I said before, LDH converts pyruvate to lactate or lactate to pyruvate according to the location and the type of reaction that we need or the cell needs, okay? So, LDH4, as we said before, is located where? Here, in the liver and the muscle. So in the liver and the muscle, we have a high demand of lactate because let's say you're working out and lactate is going to have to increase. So what happens is that the low amounts of pyruvate are directly forwarded towards lactate, or they form lactate. So the forward reaction is preferred. You see this line is longer than the reverse reaction. This line is favored over the reverse reaction because it's longer. However, LDH1, which is located in the heart, as you can see here, in the heart, lactate is not favored over pyruvate because pyruvate, can be a precursor to glucose. So the reverse reaction is favored. Thus, this line is longer than the forward reaction. Okay, so that's the difference between them. So to reiterate, LDH4 works fast, so it has a high Vmax, and it works fast as in it converts directly the low amounts of pyruvate to lactate. LDH1 converts this lactate into pyruvate, thus the reverse reaction is favored. Because pyruvate is a type of backup fuel that is needed for the heart in the form of glucose. So, what enzymes do you expect to increase during myocardial infarction and hepatitis? Myocardial infarction is a heart attack. Hepa is liver. Titis is inflammation. So, as we said before, which ones are related to the heart, LDH1 and LDH2? Which one is related to the liver, LDH4 and LDH5? Now, you're going to ask me if they're in the heart and the liver, how are they going to be elevated in the blood? Well, that's because when you have a heart attack or anything that's related to an injury to the heart, the cells are going to die. And when they die, their content is released into the blood. So when you find LDH1 and LDH2 in the blood, you have to think that the heart is injured. There is a heart injury. because normally these enzymes are not found in the blood. They are found within the heart cells. But when you see them in the blood, that means that these heart cells have been injured, and thus the enzymes have escaped from the heart and now they're in the blood. So for myocardial infarction, you're going to see LDH1, LDH2. For hepatitis, it's going to be LDH4 and LDH5. Now, I'm your patient. I come to you with, or you figure out that I have hyperlipidemia. What is hyperlipidemia? So hyper is increased, lipid is fat, and emia is in the blood. So I have increased fats in my blood. My body is not breaking down these fats. So you measure something called LPL, or lipoprotein lipase. This enzyme is normally found in the blood, okay? It's normally found in the blood. But what you measure is the levels of LPL. Is my LPL levels, or are my levels decreased or increased? In this case, if I have hyperlipidemia, of course, they're gonna be abnormal. Even though this enzyme exists normally in my blood, but since you found out that I have hyperlipidemia, you think that there's an abnormal level of LPL. What the difference is between these two cases, this one and this one, is that these enzymes are not normally found in the blood, and they indicate that there is tissue damage. But these enzymes, acetylcholinesterase, LPL, and blood clotting enzymes, they are normally found in the blood. You just measure their levels. If they have abnormal levels, then you're going to have something like hyperlipidemia, hypolipidemia, and so on. As a doctor, how can you benefit from enzymes? So basically, let's say your patient has, you suspect that your patient has glucose in their urine. So you take an enzyme called glucose oxidase, okay? If there is glucose in their urine, glucose is gonna react with glucose oxidase to form hydrogen peroxide. When hydrogen peroxide interacts with peroxidase, a color is gonna form in the urine, and this is gonna be positive. So maybe your patient is gonna have diabetes. Now, these are examples of enzymes that are related to certain diseases and certain tissues. For example, ALT, an elevation in ALT, if you see in the blood tests, then you may think of viral hepatitis or anything that disrupts the membrane of the liver. When you see amylase and lipase, you directly think of pancreas because that's where they're secreted from. LDH, as we said before, GGT is also related to the liver, and so on. I want you to understand these. I don't want you to memorize all of them right now. It's still early for you. But if you want to, please go ahead. And if your professor told you that you should know all of these, then you should. But for me, I know these, of course. By the time you're in my class, you're gonna know these by heart. But for now, I just want you to understand. I want to make sure that you understood everything that I said. And if there is anything that you haven't understood, please let me know and I'll explain it to you. And I hope this really helped you guys. I know it's a very complicated topic, but these are the basics in biochemistry, and I want you to treat biochemistry as if it's a building block. Because if your base is not good in biochem, you're gonna struggle when you go to upper classes. Because everything in biochem is related. It's not like physiology or anatomy. Today we're studying the heart, tomorrow we're going to study the stomach. Everything is related in biochem, and if your base is not strong, you're going to suffer a bit in the future. And I really hope that this was beneficial for you.